In 1349, the Black Death reached Erfurt, a prosperous city in central Germany. Fear turned into accusation. The city's Jewish population was attacked, murdered, and expelled. Homes were seized. Property changed hands. The community that had lived there for generations was almost erased in a single year. More than 6 centuries later, archaeologists opened part of Erfurt's medieval Jewish cemetery during a salvage excavation. From those graves, scientists recovered enough DNA from 33 individuals to test a question that historians had argued about for centuries. Where did the Jews of Central and Eastern Europe actually come from? The researchers expected the dead to resemble modern Ashkenazi Jews. They did, but that was not the discovery that shocked them. The people buried in Erfurt carried the same founder lineages, the same unusually shared stretches of DNA, and even several of the same disease-associated variants found among Ashkenazi Jews today. The population bottleneck that shaped millions of later lives had already happened before the 14th century. Yet the cemetery also contained something modern Ashkenazi populations no longer show as clearly. The medieval community was divided into at least 2 genetically different groups. One carried more ancestry related to Eastern Europeans. The other showed stronger connections to Middle Eastern and Southern European sources. They were buried in the same Jewish cemetery, lived under the same religious identity, and belonged to the same historical world. But their genomes revealed that Ashkenazi ancestry had not formed in one place through one event. The mystery was not solved by finding one origin. It was solved by proving that every simple origin story was wrong. For generations, 2 competing explanations dominated the debate. One claimed that Ashkenazi Jews descended almost entirely from ancient Judeans who left the Levant, settled in the Roman world, and moved gradually into the Rhineland and Eastern Europe while mixing very little with outsiders. The other argued that most Eastern European Jews descended from the Khazars, a Turkic-speaking empire whose rulers adopted Judaism in the early medieval period north of the Caucasus. Both stories promised a clean answer. The genome delivered a mixture. Modern whole genome studies consistently place Ashkenazi Jews between Middle Eastern and European populations. They share substantial ancestry with other Jewish diaspora groups and with populations from the Levant and surrounding regions. They also carry a large European component, most of which genetic models trace to Southern Europe, with smaller later contributions from Eastern or Western Europe. The exact percentages vary according to methods and reference populations, but the broad pattern remains stable. Ashkenazi ancestry is neither purely Levantine nor mainly Khazar. It formed through contact between Jewish populations of Middle Eastern origin and Europeans, followed by a severe founder event and centuries of relative endogamy. That sounds simple when stated at the end. Reconstructing it required scientists to read several different clocks hidden inside the genome. The Y chromosome follows one narrow paternal line passed mainly from father to son. Mitochondrial DNA follows one narrow maternal line passed from mothers to all children, but continued through daughters. These markers can reveal the history of particular male and female founders, but each represents only one branch among thousands in a person's family tree. Whole genome DNA combines ancestry from nearly all recent ancestors and is far more useful for estimating mixture, population size, relatedness, and the timing of demographic events. Early studies often relied heavily on haplogroups. They found that many Jewish paternal lineages were shared with or closely related to lineages common in the Middle East. Haplogroups such as J1, J2, E1b, 1b, and several branches of G and R appeared in Ashkenazi and other Jewish populations. This supported historical connections to the Levant and the broader Eastern Mediterranean. But paternal lineages were never purely Middle Eastern. Ashkenazi Levites, for example, carry a famous branch of haplogroup R1a. Because R1a is common in Eastern Europe, some researchers originally suspected a Slavic or Khazar source. Whole Y chromosome sequencing later showed that the dominant Ashkenazi Levite branch, R1a M582, has its closest diversity and likely deeper origin in the Near East, rather than among common Eastern European R1a lineages. The lesson was immediate. A haplogroup label alone is too broad. Two men can belong to R1a while their branches separated thousands of years ago in different regions. The meaningful history lies deeper in the tree. Maternal ancestry became even more controversial. About 40% of Ashkenazi mitochondrial lineages descend from 4 major female founders. Earlier work proposed Near Eastern origins for those lineages. A detailed 2013 study argued that all 4 major founders and most minor maternal lineages had deep European origins, suggesting that local European women entered early Jewish communities, likely through conversion or intermarriage. That conclusion became famous because it created a dramatic story. Middle Eastern men arrived in Europe and formed families with European women. The evidence supports substantial European maternal ancestry, but the exact picture is not as settled as viral summaries claim. Mitochondrial lineages are rare, ancient, and unevenly sampled. Determining where a branch entered the Ashkenazi population can be difficult when its closest relatives are missing from modern databases. Some maternal founders may trace to Europe, some to the Near East, and others remain uncertain. Whole genome evidence proves extensive European admixture without requiring every Ashkenazi mother to have come from one region. This is why the strongest answer comes from autosomal DNA. When researchers compare hundreds of thousands or millions of markers across the genome, Ashkenazi Jews form a recognizable cluster. They are close to one another because of shared ancestry and the founder event, yet they also sit genetically between populations of the Eastern Mediterranean and Europe. Other Jewish populations such as Sephardi, Italian, Syrian, Iraqi, Iranian, Kurdish, Georgian, and North African Jews form related but distinct clusters shaped by their own migrations and mixtures. The shared component among many Jewish groups supports ancestry connected to populations of the Levant and surrounding Middle East. The differences show that diaspora communities absorbed ancestry from the regions where they lived. Ashkenazi history therefore begins before Eastern Europe. Jewish communities existed across the Mediterranean centuries before the destruction of the Second Temple in 70 CE. Rome, Alexandria, Asia Minor, Greece, southern Italy, and other cities contained Jewish populations formed through migration, trade, slavery, forced movement, conversion, and family growth. The Roman Empire connected these communities through roads and sea routes, while also subjecting Jews to war, taxation, displacement, and changing legal status. Genetic models cannot point to one Roman town and identify it as the birthplace of all Ashkenazi Jews, but they repeatedly indicate that most European admixture occurred from sources related to Southern Europe. A 2017 analysis estimated that roughly 60 to 80% of the European component in Ashkenazi genomes came from Southern Europe, with the remainder likely connected to Eastern Europe. Because European ancestry itself forms around half or more of the genome, in some models, Southern European contribution was substantial. The researchers also found evidence for more than one mixing event. The larger Southern European-related event appears to have occurred before the severe medieval founder event. A smaller Eastern or Western European-related contribution entered later. This sequence fits a population that formed partly in Mediterranean Europe, moved north into Central Europe, passed through a major bottleneck, and then expanded east while receiving limited additional local ancestry. But genetics does not yet reveal one exact route. The traditional historical model emphasizes Jewish movement from Italy into the Rhineland, where communities appeared in cities such as Mainz, Worms, and Speyer. Other scholars argue that Ashkenazi formation involved additional centers along the Danube, northern Italy, France, Byzantium, and Eastern Europe. Yiddish itself contains a Germanic foundation, with Hebrew, Aramaic, and Slavic layers, reflecting cultural history rather than a simple genetic map. Language cannot settle biological origin, but it reveals the world in which the population formed. By the 10th and 11th centuries, identifiable Ashkenazi communities existed in the Rhineland. These communities developed religious institutions, legal traditions, schools, family networks, and local customs that became foundational to Ashkenazi Judaism. They lived in close contact with Christians while maintaining communal boundaries through marriage practices, religious law, diet, education, and worship. Then, violence struck repeatedly. During the First Crusade in 1096, Jewish communities along the Rhine suffered massacres, forced conversions, and destruction. Later centuries brought expulsions, blood libel accusations, economic restrictions, local attacks, and plague-era pogroms. Communities disappeared from one town and reappeared elsewhere. Families moved east toward regions where rulers sometimes offered protection, settlement rights, or economic opportunities. It is tempting to connect the genetic bottleneck to one famous massacre. The DNA does not identify one event. A 2014 whole genome study reconstructed an effective founder population of roughly 350 individuals around 25 to 35 generations before the sampled modern population. Effective population size does not mean only 350 Ashkenazi Jews were alive. It is a genetic estimate describing the number of ancestors whose reproductive contribution created the observed diversity. The real census population could have been larger, while only a smaller part contributed strongly to later descendants. The bottleneck may reflect a combination of persecution, disease, migration, small community size, unequal family growth, and isolation. It may also represent several contractions compressed into one statistical event. The Erfurt genomes transformed this abstract model into human evidence. The 33 individuals dated mainly to the 14th century. They were genetically similar to modern Ashkenazi Jews, demonstrating that the major ancestry sources and founder events were already established. 1/3 carried a mitochondrial lineage common among modern Ashkenazim. 8 carried pathogenic variants known today in Ashkenazi-associated medical genetics. Long runs of homozygosity showed that some individuals' parents were related at levels expected within small or structured communities. But the Erfurt population was not genetically uniform. Researchers found evidence for at least 2 groups. One, called Erfurt EU, carried more European ancestry, especially ancestry related to Eastern Europeans. Another, Erfurt EME, carried relatively more Middle Eastern-related ancestry and showed stronger effects of genetic drift. Modern Ashkenazi Jews resemble a mixture of these medieval groups, but display less internal difference. This was one of the most important findings in the entire debate. Modern genetic uniformity had hidden medieval diversity. The Jews who later expanded through Poland, Lithuania, Ukraine, Belarus, and Russia did not necessarily descend from one identical Rhineland village. Several related Jewish populations may have merged. Communities with different proportions of European and Middle Eastern ancestry came together, shared institutions, and contributed unevenly to later generations. Over time, population growth and movement blended them into the modern Ashkenazi cluster. The Erfurt study also found close relatives buried near one another, including parent-child and sibling relationships. These were not anonymous data points. They were families living in a city where Jewish existence could be prosperous one decade and threaten the next. In 1349, during the Black Death, Erfurt's first Jewish community was destroyed. Some people were murdered. Others fled. Jews returned several years later and rebuilt a second community, only to face expulsion again in the 15th century. The cemetery preserves individuals from a population caught between expansion and repeated catastrophe. A second ancient DNA discovery came from Norwich, England. 6 individuals recovered from a medieval well had strong genetic affinities with modern Ashkenazi Jews. Several were closely related, including young children. Historical context and radiocarbon evidence suggest they may have been victims of anti-Semitic violence in the late 12th century. Their genomes carried disease-associated variants that later became common enough to be recognized in Ashkenazi medical screening. This showed that parts of the founder effect were already present hundreds of years before modern genetic records. The disease variants are often misunderstood. Tay-Sachs disease, Canavan disease, familial dysautonomia, Gaucher disease, and certain BRCA1 and BRCA2 variants occur at elevated frequencies in some Ashkenazi populations because of founder effects and genetic drift. They are not evidence of biological weakness, punishment, or one ancient racial trait. When a small population expands rapidly, variants carried by a few founders can become common by chance. The same process can increase harmless or beneficial variants. Modern carrier screening programs have dramatically reduced the incidence of some severe recessive disorders by giving families information before pregnancy. Medical genetics became one practical consequence of reconstructing population history. But ancestry is not a diagnosis. Not every Ashkenazi person carries a founder variant, and people outside Ashkenazi communities can carry the same conditions. The founder event also explains why 2 apparently unrelated Ashkenazi Jews often share more identical DNA than 2 random people from a large, outbred population. Long shared segments pass through many descendants because the ancestral pool was small and later population growth was rapid. By the early modern period, Ashkenazi communities had expanded across Eastern Europe. Poland-Lithuania became the demographic center of the Jewish world. Yiddish developed regional dialects. Towns and cities supported synagogues, schools, rabbinical courts, charitable organizations, craft networks, and trade. Jews were never completely sealed from surrounding populations, but marriage within the community remained common. Genetic studies detect Eastern European-related ancestry, but it is a minority of the European component rather than the foundation of the population. The amount varies between individuals and models. Medieval Erfurt demonstrates that Eastern-related ancestry was already unevenly distributed before modern Eastern European Ashkenazi populations became genetically more homogeneous. This is where the Khazar hypothesis must be judged carefully. The Khazar Khaganate was real. It controlled territory between the Black and Caspian Seas, participated in major trade networks, and had a ruling elite that adopted Judaism. The scale of conversion among ordinary Khazars is uncertain. When the state declined, some Jewish Khazars may have joined other Jewish communities. Genetics cannot prove that no Khazar individual ever entered Ashkenazi ancestry. What genome-wide studies reject is the claim that Ashkenazi Jews descend mainly from Khazars or from a large Caucasus population replacing a Levantine origin. Ashkenazi genomes show stronger affinities with other Jewish populations, Southern Europeans, and Levantine or Eastern Mediterranean groups than with populations expected under a primary Khazar model. A 2013 genome-wide comparison found no evidence of a significant contribution from the Khazar or North Caucasus region. The absence of securely identified ancient Khazar genomes limits absolute certainty. Modern Caucasus populations are imperfect substitutes for a medieval empire that contained many peoples. A small or moderate contribution could be difficult to isolate after centuries of mixture and drift. But the major origin version of the Khazar theory does not fit the accumulated data. Neither does a pure Levantine model. Ashkenazi Jews share deep ancestry with Jewish and Middle Eastern populations, but a large fraction of their genomes came from Europeans. The European contribution was not one accidental recent addition. It entered early enough to become part of the founding population. The best-supported reconstruction begins with Jewish ancestry connected to the Levant and Eastern Mediterranean. Diaspora communities formed across the Roman and post-Roman Mediterranean. Substantial mixture occurred with Southern Europeans, possibly through conversion, intermarriage, slavery, adoption, or the incorporation of local families into Jewish communities. Related groups moved into Central Europe. A severe founder event reduced genetic diversity. Additional Eastern European ancestry entered before and after that event. Several medieval subgroups merged and expanded eastward. Every sentence in that reconstruction contains uncertainty. There may have been more than 2 admixture events. The bottleneck may have occurred over a longer period. Different communities followed different routes. Maternal and paternal lineages may preserve histories that whole genome averages blur. Jewish conversion occurred in many periods, but historical records rarely identify which converts became ancestors of later populations. Ancient DNA is beginning to narrow those gaps. The Erfurt genomes proved that the founder event predated the 14th century. They showed that medieval Ashkenazim were more diverse than modern Ashkenazim. The Norwich genomes demonstrated that Ashkenazi-associated founder variants existed in Western Europe by the medieval period. Future genomes from Italy, the Rhineland, the Balkans, France, Poland, Lithuania, Ukraine, and possible Khazar sites could reveal where major mixture and population contraction occurred. But research involving Jewish cemeteries carries serious religious and ethical concerns. Jewish law generally protects burial integrity and opposes disturbing the dead. The Erfurt project proceeded only because remains had already been excavated during construction, and researchers developed methods using detached teeth while consulting Jewish authorities and community representatives. Ancient DNA cannot be treated as more important than the people and traditions connected to the graves. Science also has limits beyond ethics. DNA does not record religion. It cannot determine whether an individual observed Jewish law, spoke Yiddish, studied Hebrew texts, belonged to a synagogue, or was accepted by a community. Jewish identity includes religion, ancestry, peoplehood, culture, law, history, and conversion. A person can be fully Jewish without matching an Ashkenazi genetic cluster, and a person can carry Ashkenazi ancestry without practicing Judaism. There is no single Jewish gene. Population genetics identifies statistical relationships among groups. It does not create a biological test for belonging. That matters because genetic research on Jewish origins is often dragged into political arguments it cannot resolve. Shared Levantine ancestry does not assign modern land rights. European admixture does not erase Jewish historical connections to the Middle East. The presence or absence of Khazar ancestry does not determine whether a community's identity is legitimate. Nations, religions, and moral claims cannot be reduced to haplogroups. What DNA can do is test demographic stories. It can show that Eastern European Jews were not formed primarily by mass conversion of one medieval Turkic empire. It can show that they were not a completely isolated continuation of ancient Judeans without European ancestry. It can reveal that their ancestors came from Middle Eastern and European populations, that mixture happened in stages, and that a medieval founder event amplified the descendants of a surprisingly small ancestral pool. The deepest surprise is not the percentage from any one region. It is how quickly a diverse medieval population became genetically cohesive. The Erfurt Jews carried differences that modern Ashkenazim largely do not. One group leaned more toward Eastern European ancestry. Another carried a more drifted Middle Eastern Southern European profile, yet both belong to the same developing Ashkenazi world. Later migration, marriage, population growth, and shared catastrophe merged their descendants. History did not preserve every community equally. Some lineages disappeared during massacres and expulsions. Others expanded dramatically. One family could have hundreds of thousands of descendants, while another left none. The modern genome is not a democratic record of everyone who once lived. It is the result of survival and reproduction under specific historical pressures. That is why the number 350 is so powerful and so easily misunderstood. It does not mean millions of Ashkenazi Jews descend from only 350 named medieval people living together in one village. It means the pattern of genetic diversity resembles the contribution of a population with an effective size of roughly that scale during a severe bottleneck. Real communities were larger, structured, mobile, and connected. The estimate captures how narrow the surviving ancestry became. After that narrowing, the population expanded extraordinarily. By the early 20th century, Ashkenazi Jews numbered millions across Europe and the wider world. Then the Holocaust murdered approximately 6 million Jews, most of them Ashkenazi, destroying communities that had existed for centuries. Survivors rebuilt lives in Israel, the United States, the Soviet Union, Latin America, Western Europe, Australia, South Africa, and elsewhere. The genome carried both expansions and catastrophes, but genetic continuity should never be confused with cultural sameness. A medieval Jew in Erfurt would not recognize every custom of a modern Ashkenazi family. Languages changed, religious movements developed. Hasidism, the Haskalah, secular Jewish culture, Zionism, socialism, migration, and modern national identities transformed Jewish life. DNA remained only one thread. Scientists did not finally solve the mystery by proving one side correct. They solved it by replacing a false choice with a sequence. Levantine and Eastern Mediterranean ancestry came first. European mixture became substantial, especially from Southern Europe. A severe medieval founder event narrowed the population. Smaller Eastern European contributions entered and varied between communities. Related groups merged as Ashkenazi life expanded eastward. The Khazar state may have contributed individuals, but it did not create the population. The pure isolation model preserved the Middle Eastern connection, but missed the European families who became equally real ancestors. In the cemetery at Erfurt, that entire history was already visible. The dead shared ancestry with modern Ashkenazim, but they were not yet genetically identical to them. Some carried stronger Eastern European connections. Others carried a more drifted Middle Eastern Southern European profile. Founder variants had already appeared. Families rested together. Above them, history moved toward plague, massacre, return, and expulsion. Their DNA did not reveal a secret race. It revealed a community still forming. That is the truth the centuries-long debate could not see. The Jews of Eastern Europe did not come from one place. They came from a chain of places joined by survival. The title says scientists finally solved the mystery, but the word solved needs precision. The broad demographic structure is now clear enough to reject the two most popular extremes. The details remain open, because every ancient genome comes from one person, one place, and one moment. 33 people from Erfurt cannot represent every medieval Jewish community from France to Ukraine. 6 people from Norwich cannot reveal the route taken by all Ashkenazi ancestors. Modern reference populations are also not perfect copies of ancient Romans, Judeans, Slavs, or Khazars. Genetic models work by comparing available groups and testing which combinations reproduce the observed patterns. When a study labels ancestry Levantine, Southern European, or Eastern European, the label describes the closest statistical source among the samples used. It does not prove that an ancestor carried a modern passport or ethnic name. Ancient populations moved and mixed too. The strongest conclusions survive changes in method. Ashkenazi Jews share ancestry with other Jewish and Middle Eastern populations. They also carry substantial European ancestry. Most models place the largest European source in southern Europe. A founder event occurred before the 14th century. Medieval communities contained more substructure than modern ones. The dominant Khazar origin hypothesis does not explain the genome. What remains unresolved is the exact geography and social process. Were the first major European mothers concentrated in Italy, the Aegean, southern France, or several Mediterranean regions? Did conversion happen mainly in late antiquity or continue through the early Middle Ages? Which Central European communities survived the bottleneck and contributed most strongly to later expansion? How much ancestry entered from Slavic neighbors before and after movement east? Each future cemetery could change part of the timeline. A first millennium Jewish genome from Italy might identify the Mediterranean mixture before Ashkenazi identity existed. Early Rhineland genomes could reveal whether the bottleneck had already begun. Medieval Polish genomes could show when Eastern European ancestry increased. Securely identified Khazar period Jewish genomes could finally test for limited connections that modern data cannot isolate. The medieval evidence also warns against reading present-day genetic uniformity backward. Communities in Germany, France, Italy, Bohemia, and Poland may once have differed more sharply than their descendants do now. Persecution could eliminate one branch, migration could combine two branches, and rapid growth could make a third disproportionately influential. A modern population cluster is therefore not a photograph of one founding village. It is the surviving average of many communities whose contributions were unequal. That is why Erfurt itself matters. The cemetery captures Ashkenazi history before later expansion smoothed away part of its internal structure. It shows that religious unity and genetic diversity could exist together, and that the population recognized today was assembled from related groups rather than copied unchanged from a single source. The mystery was never hidden in one haplogroup. It was hidden in the order of events. Until then, the most honest answer is layered rather than absolute. Ashkenazi Jews emerged from Jewish populations with deep Middle Eastern ancestry, European families who joined them, medieval communities narrowed by demographic crisis, and Eastern expansion that added smaller local contributions. The result became genetically distinctive without ever becoming biologically pure. That is not a compromise between two theories. It is the history written across the whole genome, across